Hello, everyone. Thank you for coming to this session. So how we're going to structure this, this is primarily a hands-on session. So I have a few slides to introduce Pine and explain the motivation, why it exists, and in broad terms, what it does. And then we'll dive in. So I prepared a Jupyter notebook in the eBrains lab, which I'd like everyone to kind of work through together. And we'll take it slowly and make sure we understand each step in the tutorial. And if you have any questions, please either speak up here in the Zoom. That's easiest because then I won't miss your question, or put a question in the Zoom chat. Or there's also the eBrains chat. I put a link to that, so you can also ask questions in the eBrains chat application. So yeah, getting started with the presentation. So Pine is a layer which sits on top of simulators to provide a unified interface to different simulation tools. So if you look on the ModelDB database, they have a page which lists models according to which simulation tool, which simulation environment those models have used. And you can see that, so I took this screenshot several years ago, and even then it was already a very long list of simulation tools. And this, this is potentially a problem. So obviously a single researcher, a single lab can't model everything that's of any interest. We need to build on previous work. We want to reuse and extend existing models. But doing that is made more difficult because each model is usually written for a single simulator. And so if you have a good reason to use a different simulator, you have to convert that model, translate it, and that takes a lot of time and is error prone, and it's difficult. So most models are not very reusable or even not very testable. On the right of this slide, everyone sees my slides, by the way? I guess yes, because no one says yes. On the right of the slide, you can see that certainly 10 years ago, it used to be that if you wanted to write a model for the Nest simulator, you have to use this SLY language, and you get the data in the format that Nest uses. If you wanted to write a model for Neuron, you'd have to use the HOOK and NModel languages, and again, you get data in a different format produced by Neuron. If you wanted to use Brian, you would write Python code, and again, you get a different format of data. So, as I said, there are this large number of simulators is a problem. Makes it difficult to translate models, difficult to understand people's code, and that makes it just generally slows down the field. However, it's also an opportunity having all these different simulators because each simulator is good for different things. So some are more efficient, some are more flexible, some scale up well on supercomputers, some are more user-friendly. And so it's good to have multiple ones because then you can choose the one which is most appropriate. And secondly, essentially almost all code, all software has bugs. I mean, the exception is maybe, for spacecraft, for nuclear power stations. I hope those have been thoroughly debugged, but almost all of the software has a lot of bugs, and also just hidden assumptions that about how things are implemented. And these things can be revealed by cross-checking between different simulators. So if you get the same result from your model in two different simulators, you have much greater confidence that your results are correct. Now, we can probably get the benefits of having multiple simulators without the negatives by using a simulator-independent environment for developing models. There are various approaches to that. So there are some graphical tools like Neuroconstruct, which lets you build your models build your models in a GUI and then execute them on different simulators. There are various XML-based languages or JSON-based languages like NeuroML, or you can do what Pine does and use an existing interpreted language, in this case, Python. So what Pine does essentially is instead of having to write different versions of the model for different simulators, you write a single Python script. Pine will run it for you in Brian or Neuron or Nest, or even in neuromorphic hardware like Spinnaker and Brainscales, and there will be tutorial sessions on those later in this workshop. And then in addition, you get the data out in a single format without having to have the different formats for all different simulators. So by having this common interface, common API for neuronal network modeling, you can make model sharing, model reuse much easier. You can make cross-checking and validation much easier. And you also have a platform on which you can build other tools which don't have to deal with different simulators. They can just deal with the Pine API. A quick comment on how this works. Pine does not attempt to reproduce all of the functionality of all simulators, nor does it reproduce only the kind of common core, so the dark orange in this slide. Instead, Pine tries to reproduce anything which two simulators can do. So as long as simulator 1 and simulator 3 can do it, Pine will try to do it as well. So it's this kind of central, slightly expanded orange area, which is what Pine aims to do. If you want to find out more about Pine after this tutorial, you can go to neural-ensemble.org/Pine. It's open source licensed, and we also have a Google group /mailing list where you can go to ask questions. We'll come back to that. A quick example before we get into the hands-on. This is what a Pine script looks like. So we set up our model choosing the integration time step. We specify some parameters for the neuron models, so for integrating fire models, the memory and time constant, the memory capacitance, the voltage threshold for spiking. Then we create some populations of these neurons. So here we're creating 20,000 this type of integrating fire cell, and here another 5,000 cells. And we're going to create some random noisy spiking activity. And we're going to inject some noisy current as well into our neurons. We can then connect them up. So we want to have random weights in our network. We want connectivity, which depends on the distance. So the further apart they are, the less chance they have to be connected. And we can also see that here the delays also depend on the distance. So as the distance between the cells increased, the time delay for the spikes increases as well. And we want these synapses to be depressing using the Zotix-Markram model. So if you have a burst of spikes all coming in rapid succession, then they will each have a smaller effect as time goes on. And so we put all this together. So we create a projection connecting the excitatory population to everything else using this connection algorithm, using this type of synapse type, and with excitatory connections, and then a similar connection from the inhibitory neurons to everything with the inhibitory connection type. And don't worry, we'll go through all this in much more depth in the hands-on part of the tutorial. But just to say, if you want to run this simulation with Nest, all you do is import pine.nest as sim at the start. And if you want to run it with Neuron, you just import pine.neuron, Brian, neuromorphic hardware. You get the idea. None of the code, apart from the very first line, needs to be changed. So I'd now like everyone to go to the URL on the screen. Let me copy that into the chat for you. There we go. So as I said, the next part is interactive chat. So please go to this collab. It is a public collab, and I'm going to stop sharing the slides now and move to sharing my collaborative window. So as I said, the link is in the chat. No, not that one. Why is that? Okay, that's weird.
Looks good. It's Pine Resources.
Okay, you're seeing it. So, sorry,
Yes.
I'm getting a bit confused. I'm still seeing my slide on my screen. Right. So in this Pine resources, please click on hands-on sessions and then click on simulate with eBrains workshop. And you'll come to the instruction page for the current workshop. As I said, there's some links here to some resources. So there's the chat.ebrains.eu. There's a channel about Pine, so you can ask questions there during the session, and I should get notification and I can reply to you there. You can also ask questions in the Zoom, or just orally. You can just speak out. Please feel free to interrupt me at any time if you have questions. I also put a link to some Pine documentation, to a couple of YouTube tutorials for using Pine. So today we're going to be just using Pine in the eBrains lab, but you can also install it on your own laptop or on a supercomputer. And so at least for Linux so far, we have some instructions on video form for that. If you're using Windows or Mac OS, then you should look at the documentation for the instructions. And then there's a YouTube tutorial, which is quite similar to the tutorial we're going to go through today. And then finally, if you have any problems using the collaboratory, like the lab and so on, then I put a link to the collaboratory documentation. But hopefully everyone watched Paul Cheney's presentations yesterday. You should all have a good idea of how to use the lab already. So what we have here is a notebook, which you need to copy into your own Colab workspace. Did everyone have their own Colab?
Collabs have been created for the people who selected one of the NMC sessions.
Okay.
But others may or may not have a collab yet. So it's good that you say you can create a new collab in your description.
Yes. So please, if you do have an existing collab you can use, then that's good. If you don't, then please either click here on the wiki page, or you can go to up here, click on collabs, and then click on create a new collab. So I'll give you everyone a minute to do that. If you could all maybe in Zoom put your hand up when that's done, and then I'll know I can move on. Okay, so I think we have someone new just joined. So you can get caught up. I'll just put in the link in the chat. you should go to that page where there's instructions for the hands-on session. And what we're doing at the moment is creating a collab, so each person can run the Jupyter Notebook in their own workspace. So that's line 1 of the instructions. If you scroll down, you can see that there is a link to create a collab of your own, and unless you already have one which you can use, which you can reuse. If anyone's having any difficulty, also please please let me know. Please speak up. Mathieu, Payman, how's it going? You still in progress?
No, it's okay. I have it.
You have it. Okay. So can you in Zoom? Can you put your hand up? Just that way I can see when it's. So if you have it, please put your hand up. That way I know when I can move on.
I very much assume that several of the others here have already collabs, because I recognize some.
Okay, yes. So OA, I will put again the link. What we're doing at the moment is creating a collab. So if you already have a collab to work in, so you can save any changes you might make to the Jupyter Notebook and so on, you can please use that. That's great. So otherwise, so the other thing you perhaps missed is I put a link to a wiki page which has instructions for creating a collab and doing the rest of the hands-on tutorial. Okay, so please, if you're still working on making your collab, please do so. Otherwise, can everyone put your hand down now? Okay, and so the next stage is to copy the Jupyter. Just wait for that's it. The next stage is to copy the Jupyter Notebook into your own collab. So let's do things a bit differently. Can I have everyone put their hand up now? That way I can see who's actually interactively involved with the session. And then you can put your hand down once you've achieved the next step. So please, everyone, put your hand up in Zoom. Okay, I assume that the people who haven't done so are just listening and not following along. So, okay, so the next step is to copy the Jupyter Notebook into your own collab. There are two ways you can do this. There is a kind of two-click approach, which we can try. Option A. If option A doesn't work for you for some reason, then try option B. It will certainly work, but option A should be fairly simple. So you click on the link, and then you find a collab that you want to use. I have quite a lot of collabs. So whichever collab you want to use, click on that, and then click continue, and then just click clone file, and that should copy the notebook into your new collab and open it for you in Jupyter Lab. If it doesn't work, then try option B, which is in this collab here, the Pine Resources one. Click on Drive. It's perhaps a bit slow, the collaboratory today, because everyone's busy using it. And then hands-on. And then simulate with eBrains 2022. And then in this drop-down menu here, more operations, choose copy. And again, you can navigate through to your collab that you want to use, and it will copy it there for you. And then once you've done that, once your the notebook is open in the lab, then please put your hand down. If you need to start the lab manually, then in your own collab, so not in this one, not in the Pine Resources one, in your own collab, click on lab, then select, it shouldn't matter, either the Swiss National Supercomputing Center or the Jülich Supercomputing Center. And that should start up here. You can click on this button here with the arrows pointing in all directions to expand, make it bigger, or you can open it in a separate window using the Open in Lab button. That's the wrong one. Hang on. So yes, so then in this left-hand window, you should see hands-on. Well, yeah, in your own collaborative, you should just see the right notebook. So I have to go through and find the right one. This one. So once you have this open, please put your hand down in the Zoom. And if you're having problems, please just speak up.
Hi, I'm just like have the collab right now, but I didn't get yet to this tutorials files.
Okay.
So if you could please again like let me know how to get into these files.
Yes. So in the Pine Resources collab. Here we go. Hands-on sessions, simulate with eBrain's workshop. There is an automatic tool which I see in the chat that some people are kind of helping each other out with this. Yes, so when you create your collab, I forgot to mention that, you first of all need to initialize the drive. So in your new collab, you have to click on the drive link at least once before you do this. So first of all, click on the drive in your own collab. When that's done, come back to the Pine Resources collab and try clicking on this link here in step 2 of the instructions. And as I say, you choose a collab to work with. Too many collabs. And then yes, you can, by default, it will copy it to the root directory of your collab. You can also put a subdirectory in here if you want. And then click continue, and that should copy the Qubit notebook into your own collab. And then it should, you should end up with something looking like, where are we? Find the wrong window. No. Yeah, this one. And once that's done, please put down your hand in the Zoom window so I know that everyone's ready to move on.
And just for info, I added into the online agenda the link to the page that you were showing in your collab, this description for the hands-on session today. So in case anybody missed that link, it's now in the online agenda.
Thank you. So just one person still working on that. So while that's going, just for the rest of you, please check that. So up here in the upper right of the Jupyter window, you should see the kernel you're using, and you should see eBrain's 2022-10 RC. And that is the latest eBrain's release. It's not quite official yet. That's what RC means, release candidate. But this will very soon become just 22.10. And this is the latest version of all the eBrain's tools. If you don't see that, if you see something else, then click on it. And you'll see there's a drop-down list here of different versions you can use. So 22.07 is the current official version, which will soon be replaced. But we want to use the latest version, 22.10 RC. And if you have to change it, click select, otherwise cancel.
Excuse me again, I'm sorry,
Yeah.
but when I clone the whole files, just shows me like there are drive and shared files. So which folder particularly should I find this notebook?
Are you currently in your own collab?
Yeah.
Okay, so it sometimes takes a little while for the drive to kind of catch up.
Mm-hmm.
So possibly if you wait a little while, it will appear. If not, then if you double click on shared, somewhere in the list which appears, you should find your collab. I think this is the only disadvantage of doing these virtual tutorials. When we've done these tutorials in person, then usually I have some helpers who can help people get set up and so on. So I apologize.
If we have a helper, well, I could at least go with into a breakout room and have a look at, unless you have somebody else from your team. No, there's nobody from your team. Ah, Mathieu is there.
Yeah, there are a few people from my team.
Let's create some better breakout rooms, then people can go there. Do we want to do this?
How are you getting on? Sorry, I don't know. You appear as OA in the Zoom window, so I don't know your name. But how are you getting on with?
Yeah, again, I'm just I found my collab, but it seems it's empty. So, but I have all the shared file and all the drive, everything is ready right now, but just I couldn't find what you show in the window right now.
Okay.
Like the tutorial.
Well, would you like to share your window and then?
Yeah, that will be amazing. Yeah, sure,
okay,
sure.
I'll stop sharing and then you can share your window and then we can talk you through.
So, can you see it right now?
Yes.
Okay. So, this is like the landing page that I had when I did everything. So where should I go, like for the drive or the shared?
So if you go to shared.
Okay.
Now, what was the name of the collab that you created?
It's BYNN-1. Yeah, I found it. It's here a little bit down. Yeah, this one.
Okay, so double click there.
Ah, now
one.
it appears. Yeah, it was not there. Thank you. Thank you so much.
Okay, great, thanks. Yeah, so if you stop sharing, I will share mine again.
Sure.
All right. Okay, that's good. So what we're going to do in this tutorial is we're going to build a simple network of integrating fire neurons with Pine. We're going to run some simulations of this model using different simulators, and we'll take a look at the data generated by these experiments. I'm assuming that everyone here has at least a basic knowledge of computational neuroscience. So first of all, yeah, if there's anyone here who doesn't know what an integrating fire neuron is, please speak out. I could say a few words about that if you wish. Okay, good. Otherwise, I'm also assuming that you have a basic familiarity with Python. And if you haven't, then you can still follow along because the code's already written for you. Again, I assume that most people these days do know how to use Python. There's two ways we can do this. We can do this. Everyone goes through one at a time, but I think probably it's best if I talk through. Yeah. So as I say, installation. Nothing to do here. Everything is already installed in the eBrains lab. If you do want to use Pine for your own research later, then it's fairly straightforward to install Pine, just a Python package. What is something on your own computer or on a HPC machine? What's a bit more difficult? Sometimes it's to install the simulators themselves, Nest or Neuron, or whatever. But it's not too challenging. In any case, you will find instructions for all of this in the Pine documentation, which is linked from the Pine resources page. But yeah, none of that concerns us here because we're going to run everything in the lab. So what we'll do is we'll get started by choosing a simulator, and I rolled the dice, randomly chose Brian 2. And so we can import the Brian 2 module for Pine. So import pine.brian2, and we'll call it just sim to have a generic name for it. Now, how Brian solves equations is by advancing time in small steps, and typically 0.1 milliseconds. That's kind of in biological time is long enough to give accurate results. It's small enough to give accurate results. With any model, if you do find that your results change when you change the time step, then your time step is probably too large. So it might be interesting to try to rerun the same simulations with a time step of, say, 0.01 milliseconds and just see if that changes your results at all. Then rerun the time step of 1 millisecond and see if that changes your results. I expect that it will. So Pine comes with a selection of integrating fire models of various kinds. So our simple integrating fire. There are also more complex examples like the Itzkevich model, the adaptive exponential integrating fire model. But we're going to use here a very basic one, so-called IF-Kerr-Exp model. So IF stands for integrating fire. Kerr means that the synaptic response is a change in the synaptic current only, so not the conductance, but just the current of the synapse. And then the exp means that this is a decaying exponential function. So when there's a spike, the postsynaptic current jumps up and then decays down exponentially. And we're going to set some parameters of this model. So here you can see cell type. So I forgot to say we should have executed this cell. So either click on run, or you can do control enter. So import pine.brian2 as sim, set the neuron up, and then the next thing is to set the cell type. And here we're setting the resting membrane potential to minus 65 millivolts, the threshold voltage for a spike to minus 55. After a spike, we will reset the membrane potential again to minus 65. There will be a refractory time. So after each spike, there's no change in the voltage for 1 millisecond. The membrane time constant is 10 milliseconds. The membrane capacitance is 1 nanofarad. And we're going to inject an offset bias current of 1.1 nanoamps. So Pine uses a consistent set of units. It's always millivolts for voltages. It's always nanofarads for capacitances. It's always nanoamps for currents. So that's our cell type. And now we're going to take this cell type and create 100 neurons of this type. So here we create a population. So sim.population, 100 is the number of neurons. This is the cell type. And then we can give it a label if we wish to distinguish it. What we're going to do then is record the membrane potential from these neurons. So from our population of neurons, we want to record voltage. And then we're going to run a simulation for 100 milliseconds. So if we run that cell, it will take a few seconds to run. And it tells us at the end that we have indeed run it for 100 milliseconds. You can ignore these warnings, which is Brian telling you what it's doing. So we now have some data stored inside our network. Now we want to look at this data. So let's start by enabling plotting with matplotlib. And we're going to use some built-in Pine utilities for plotting, which make this a bit less verbose. So we import the figure and the panel classes from Pine utility plotting. And now we're going to get the data using this command here, population1.getData. And then we're going to drill down into the first segment and filter out for signals which have the name voltage, name V, which matches what we recorded up here. And then in our figure, we have one panel which has the data which we recorded and some labels for the axes. And if you execute that, you should see a figure like this. If anyone sees something different, like an error message, please let me know. Okay, so I assume I can move on. As I say, at any time, if you see something different from what you see on my screen, please speak up. And so what we're seeing here, of course, is that because we've injected a bias current into these neurons, or this neuron, neuron 0, the membrane voltage increases and reaches threshold. Now, in a real neuron now, you'd see a big spike up here. Because this is an integrated fire neuron, the spike is kind of implicit. It has zero width. So it goes like, imagine it goes up here and then comes back down to the reset voltage, takes a millisecond in the refractory period, and then starts to go up again for the next spike. So what we saw was a rather complicated command. So population1.getData.segment0.filter name equals v.0 bracket 0. That looks a bit complex at first glance, so I think it's worth spending some time to explain what we're seeing here. So Pine is using the NEO library for representing data. NEO is a library which makes it easy to share data among different tools. So for example, I think we have the Elephant tutorial later this week. Elephant is also based on NEO, so any data you create with Pine is very easy to analyze with Elephant. NEO contains data in analog signal and spike train objects, among others. And these objects are very similar to NumPy arrays, but they have extra metadata attached to them, like the units, the sampling rate, and so on. And so this data, like analog signal and spike train, we store we can group them together inside a container called a segment. So each segment can contain multiple signals, multiple spike trains, various other things as well. And then you can group multiple segments together into a block. So let's suppose you run a simulation, you change some parameters, run a second simulation, then that would be two different segments inside your block. So in this case, we just ran a single simulation. So we have one block and one segment. So what we're seeing here is get data gives you a block. The block has a list of segments. We want to take the 0th segment. And then this segment then contains potentially a lot of things. So we're going to, as a shortcut, just filter it to get anything which has the name V. And that will give us a list of signals. And we take the first signal. There should only be one signal, or the 0th signal, but there should only be one. So what we end up with is an analog signal, which contains channels for each neuron. So each neuron has a separate channel in this signal object. So it's an array which has, however many, 1,000 time points. So it's 1,000 by 5 is the dimensions of our NumPy array. So that's where this rather long name, this rather long command comes from. So it's a bit boring seeing like 100 identical neurons. So here we plotted the first neuron, as you can see. This was all the time points, but from the first channel 0 in our 2D array. How about instead we plot all 5 neurons. So here we're going to plot all 5 neurons on the same axes. And yeah, it looks pretty similar because of course they're all identical neurons, so they all have exactly overlaying responses. So that's kind of boring. Let's see, did I execute that? No, let me execute that one. Okay, yeah. So of course in nature, every neuron is a bit different. So what we can do is we can add some randomness. So each neuron in our population will now be slightly different from the others. Just to do that, let's start a new simulation. So we reset the simulator. And now we're going to use some randomness. So from the Pine random module, we're going to import the NumPy random number generator and a class called random distribution. And we will initialize the RNG with a seed. So here I encourage you to change this. Let's change this seed to something else. So everyone has a different seed. Make things a bit interesting. And now we're saying that our resting memory potential, instead of being just a single value of -65, is now going to be a normal distribution with mean of -65 and standard deviation of 1 millivolts. And we're using this NumPy RNG we created earlier. Similarly, the threshold for spiking is going to be also from a random distribution, and the reset also from a random distribution. All the other parameters, we will keep the values they had before, although there's no reason you couldn't also randomize those ones. And now again, we just create our population, same way we did before. And just to check it's worked, we can take a look using the population.get method. We can take a look at the vrest parameter, and we can see that it is, we do indeed have random numbers for these for this parameter. In comparison, the tau m, we just kept the same value. They should all have the same value, which is 10. So again, let's run our simulation and plot the results. And now we can see that we do indeed have some randomness. So our neurons are all spiking at slightly different times, resetting to different values, and so forth.
Andrew, just a quick question.
Yeah.
On your screen, the tau m is a full array. On my screen, it's only a single 10. Is this getting of the parameters related to the simulation backend?
Are you using something other than Brian?
I, of course, use Nest. Yes.
Yes. So apparently, yes, that's actually a bug you've discovered. So it should give the same. Of course, it doesn't matter under the hood. All neurons have the same value, which is 10. Pine does try to be clever, and if they're all the same, then it tries to only show a single value. But obviously, there is a bug here, and it should be showing probably a single value for Brian as well. So yes, thank you for that bug report. I will fix that.
Yeah, thanks a lot.
Yeah, so we've run that. Yeah, so but at least you should see the same result. That you should see that all each neuron is now given slightly different results. All right, any other questions? Okay, then we can continue. So far we have a population of neurons. They're all slightly different, but they're not connected to each other. So they're only spiking because we're injecting current into them. So now let's add a second population, same size as the first, but in the second population we'll turn the current off so that they don't automatically fire. They will only fire spikes if they receive synaptic input. So again, in this cell we're going to reinitialize the simulator and now create two populations, population 1 and population 2. They're the same cell type in this case. They could certainly be different, using the same parameters as previously. But now we will again use another method, population.set, to turn off the bias current in the second population. So we now have two populations which are the same, except for some randomness, and that the second population has no bias current. And we're also going to record V from both of those populations. And now we're ready to connect them up. Now, there's lots of different ways we could do this. We could connect everything to everything else, or we could connect them randomly in different ways, or we could connect them randomly but with a specific dependence on distance between neurons. So you might see in this figure here, neurons which are close by are more likely to be connected. This neuron doesn't connect to things which are over here, for example. Or we can say, okay, I know exactly which neurons should be connected to everything else. So I'll give you a list of the presynaptic index, the postsynaptic index, and this is how I want you to connect the network. So just as Pine provides a variety of neuron models, as I showed just now, it also comes with a range of connection algorithms built in. So all these ones here, among others, and you can also write your own connection algorithm for Pine, just using Python code in a way which will work with any simulator. And that's, I won't show that here today, but that's reasonably straightforward to do. So if you find that the Pine built-in methods are what you want, it's quite straightforward to write your own Python class, which will do this. So in Pine, we call the group of all the connections between one population and another, we call that a projection. And to create that, we need to tell it, okay, which neurons am I connecting from? Which neurons am I connecting to? What's the algorithm I'm going to use to wire these up? And what's the synapse model that we're going to use? So here we're going to use one of these Pine built-in connection algorithms called the fixed probability connector. So for each possible pair of neurons, so like 0 to 1, 0 to 2, 0 to 3, 1 to 2, 1 to 3, and so on, we generate a random number between 0 and 1. If it is less than 0.5, we create the connection. If it's more than 0.5, we don't. So this is going to connect, on average, half of our neurons to each neuron will connect to half of the other neurons. We're going to use the very simplest model of a synapse, where the weight is always the same. So each time you get a spike, you get an increase in weight of a constant amount and a constant synaptic delay. Again, there are more complex models with depressing or facilitating weights or spike timing dependent plasticity. There are various models you can use. We'll just use the simple one today. And then we put this together by creating a projection. So sim.projection, specifying the presynaptic population, population 1, the postsynaptic population, population 2, the wiring method, and the synapse type. And then by default, because we didn't specify it, these will be excitatory connections. We could also specify other connection types, but by default, they are excitatory. So where did I stop? So I executed those. So I need to execute these lines and run the simulation. It's a bit longer because this time Brian is having to deal with all the spikes being transmitted. Okay, that's done. So now let's extend our figure. So we can add a second panel to our figure, and in which we're going to show the data from population 2. So here is what we have. Execute that to get the data from the populations, and then this to plot them. And we should now see that, as before, the first population just spikes because it's having some current injected. Then each time there's a spike, there's a postsynaptic potential, and that's making some of these neurons spike in turn. There's quite a high weight, so a single spike is enough to make the postsynaptic neurons spike in turn most of the time. Okay, so now, Dennis, you're going to have to do this in reverse. We've just run this network with Nest. So most of us are running it with Brian. We're now going to do the same thing with Nest. You can do the reverse, or you could choose Neuron, for example. So essentially what I've done is I've gathered all of the code from the previous cells into a single cell. So now putting this all together, this is what our model looks like. We import our simulator as sim, pine.nest, pine.neuron, pine.brian. We import our other classes we need, the plotting classes, the random classes. We initialize our simulation. We create the cell parameters. We create our populations. We record neurons. We connect them all together. We run our simulation, and then we plot the results. So the same thing again, much faster. Nest is in general faster than Brian. And so you should all see this, which is essentially, should be identical or almost completely identical to, except that the seed I didn't change here. So I changed it above, I didn't change here. So you will see slight differences, but if you use the same seed, you should see the same results here and with the Brian results. So quick exercise before we move on. So I'd like all of you to now try to run this same model with a third simulator. So if you used Brian and Nest already, then try and run the same thing with Neuron. And then once you're done with that, please put your hand up or put a thumbs up, and we can move on to something, to the next part of the session.
I have a quick question.
Yeah.
So I've added the same seed, and results seem to look very similar, though one of the neurons seems to have an extra spike somewhere in between. Is this to be expected, that there is a difference between the simulators for random number generators?
There should not be any difference in the parameters or the connectivity. So we could go into that and take a look. But I suspect the reason is just that as you know, spiking neural networks are very sensitive to initial conditions. They are close to chaotic systems, and so a very small change due to floating point representation or due to slightly different algorithms which are used to solve the differential equations. These very small changes are amplified by the fact that there's a spike. So if you have a very small difference such that one neuron almost reaches threshold but then doesn't, and then the other one just reaches threshold, then you'll get a spike. Until then, they were almost identical. All of a sudden, you have a spike in one and no spike in the second one. I suspect that's what happened here. It's possible there's a bug in the random generation, but normally the reason for using the Pine random number generator, the NumPy RNG, is that it should take care that you get the same random numbers in all backends. By default, you can turn that off because that has a performance hit. So if you're happy to get different random numbers in different backends, you can turn off this feature, which gives you the same numbers in the different backends. But by default, that's what should happen.
Yeah, so I just found a line of warning that might also be related to this. So when running the first part with the Nest backend, it actually tells me Nest does not allow setting the initial value for isync x and isync inc inhibitory.
Yeah, that is true. That is not a problem in this scenario because the default value in Pine and in Nest is 0, and we're not changing that default. So in this, if you wanted to set a different value other than 0, it would be a problem. But in this case, it's not a problem because we're using 0 in all the backends.
Okay.
That's a feature request from Nest, by the way, also to allow us to set this value.
Yeah, perfect. Thanks a lot.
Okay, so I think most people have, most people who've been putting their hands up so far have done so. Would anyone like to speak up and say, hold on, something's not working for me? Otherwise, I think we'll finish now with the hands-on part, which we got through pretty quick. So I deliberately kept it quite short because sometimes, other tutorials, there's been a lot more problems to do with installing the software and so on. I think probably now that we're using the eBrain standard software, I can actually assume that things will go much more smoothly. So perhaps I will extend this tutorial next time. But what I'll do now is go back to my slides and perhaps put some of what we've seen in the hands-on into context and do a bit more context and give you a bit more background information about Pine. So you should now be seeing my slides again. Okay, good. Yeah, so just this is partly a recap of what we've already seen in the hands-on part. So in the API, we can break it down into neuron and synapse models, populations, connectivity, and then recording and data handling. So Pine comes with so-called standard models. So these are models which are available in at least 2 simulators, often in all of them, but at least 2. You can also, if you wish, use native models. So you can use models which only work with one simulator. And then you can also use some tools like 9ML and NeuroML, which let you specify neuron models using a different language. And you can compile those and use those in Pine. So briefly, Pine has a list of standard models. If you do the list standard models function in your simulator, it will tell you which ones you have. This list will be slightly different for Brian, for Nest, and for Neuron, because there is not a complete overlap. And as you've seen, you create your cell type by using this class with your parameters, and the synapse type is the same. So I showed you the static synapse. There's also the Solix Markram synapse, again, which has slightly more parameters. And what's happening under the hood is that Pine is basically converting the parameters and converting the neuron models to the appropriate name. So if we take the Pine IF current alpha model, it has a parameter called vrest, for example, the resting membrane potential, which is in millivolts. Now, with the neuron simulator, this model is called standard IF. This is part of Bride with Pine. It also has the parameter name vrest, and it also has millivolts. The equivalent model in NEST is called IAF-PSC alpha, and the parameter is called E_leak. So this is the leakage voltage. It's also in millivolts. But if we look at the capacitance, for example, it has a different name in every simulator, and it has different units in, say, Pine and Neuron compared to NEST, where it's picofarads, not nanofarads. And actually in Brian, it's just farads. And so what Pine is doing under the hood is kind of translating all of these names and all of these parameter units to the appropriate parameterization for each simulator. It's sometimes a bit more complex than that. Sometimes models use different parameterizations, but generally it's along these lines. You can also use native cell types. So here we're using in NEST, we import these functions: native cell type, native synapse type, and we can use these models which are built into NEST, HT neuron and Poisson generator. This of course will only work with NEST. It won't work with any other simulator, Neuron or Brian. But if you're otherwise using, if you like Pine otherwise, or you let's say you have a NEST model which you want to convert to be completely independent, then you can start off by converting just the connectivity and leave the neuron types the same using this native model functionality. And it's the same for the plasticity models and the synapse models. So here you can use a built-in NEST model called STTP synapse in Pine. Neuron, it's a bit more complex to do this. You have to define a couple of small classes. Well, if this is the model you're trying to wrap, then you have to define this rather simple class to make this model usable with Pine. So then there's also model description languages like NeuraML, NineML, or NestML. There's a NestML tutorial I think happened earlier today. We don't yet support NestML in Pine, but that's planned. We do support NineML, which gives models for Neuron and for Nest. And so essentially, you can define your model using the NineML language, which can be saved to XML format, for example. And then Pine can use this NineML model under the hood. What's happening under the hood is it's compiling this either into C++ code for Nest or for an N model file for Neuron. But Pine does this for you, and then it uses this model behind the scenes. Moving on to then populations, what we've seen so far in this morning was the population class. This is a bunch of neurons which might have slightly different parameters. So we saw that we can have random variability, but each model is of the same type. It's using the same equations. And so we can have two different populations using either the same equations or different equations, so two different models. We can also then break things down. So there's a class in Pine called population view, which lets you focus on just a subset. So let's say we wanted to inject current into only a subset of the neurons. Then you can specify, okay, population view of the first 4 neurons or a random sample of neurons, and I inject current. I set the parameters differently for those neurons. And then if you want to group things all together, like multiple populations, or let's say this population and then just this part of this population, you can group those with an assembly. And the motivation for this is, for example, let's say you're doing a model of cortex. You might have a different population for each cell type. So this might be pyramidal neurons in layer 2, 3. This might be interneurons in layer 4. And this is spiny stellate cells in layer 4. And this is pyramidal cells in layer 5, let's say. And then we also want to specify columns, so cortical columns. And so we can specify that using an assembly, for example. So this assembly will group, this will be a population view. All of these here will be population views. So we can have an assembly which cuts across the layers in a cortical column. So this combination of population view, and assembly lets you give a kind of a rich structure to your network model. As I said, population is n neurons, all of the same type. You create a population view just using normal Python indexing syntax. So this is the first 80 neurons. This is every second neuron. This is a random sample of neurons. And then you can connect these things by adding them together. So if this is a population or a population view, you just add them together, and this resulting layer 4 is an assembly. We've seen that you can make parameters random using the random distribution object. You can also set them all to a single value. You can also specify a function, which will take, so if you have 1,000 neurons, then this index going from 0 to 1,000, you can specify whatever function you want. So you can have the parameters of the different neurons in a single population all be different based on some function. Or you can set them to using an array. So if here we have n neurons, then we can specify these linearly across the population using a specific array. So all of these options exist for parameterizing your neurons. Moving on now to connectivity. As I said, as we saw in the notebook, a projection is the set of all connections between two populations. And it's not just populations. You can also use, anywhere I said population, you can also use population view or assembly. And as I said, we specify the presynaptic, the postsynaptic population, the connection algorithm, and the synapse type, and then together with the type of the postsynaptic neuron optionally. And we can also specify some other things. So, for example, you can specify whether you want space, when you're calculating distances, whether you want to calculate them in like all 3 dimensions or just, let's say, 2 dimensions in the plane. Or if you have a one-dimensional network, like in a ring structure, maybe just 1 dimension for space. So you can control all this in a very fine-grained way. As I said, each connectivity algorithm is encapsulated in a Python class. This is the library which already comes built into Pine, and it's not too difficult to write your own. And then finally, recording and data handling. As I said, Pine is built on Neo, which is also used by lots of other tools in computational and systems neuroscience. And all the data objects are contained inside segments, which are contained inside blocks. And as I said, this is all based on NumPy. So the base is a NumPy ND array. We then use the quantities library, which adds units to this. So for example, if you try to, let's say, add an array in millivolts to an array in microvolts, it will do the unit conversion for you automatically. And if you try to add an array in millivolts to an array in nanoamps, it will produce an error. So you'll get it stops you making mistakes of that kind. And then Neo in turn wraps quantities and adds extra metadata like what's the sampling rate of this signal, what's the start time of the signal, and so on. So this is an example of using multiple segments. So here, this is just a single neuron. So 1 neuron, Hodgkin-Huxley. I'm going to inject a step current into it. So I didn't show you this in the hands-on, but you can specify various current sources. So this is just a step on in the plateau and a step off of the current. Steps on at 20 milliseconds, stops off at 80. And we're going to inject this current into this cell. Record voltage. And now we're going to run a bunch of simulations. So I'm going to change the input current from minus 0.2 nanoamps to plus 0.2 and run a simulation each time, and then reset the simulator back to the starting point each time. And so each time we reset the simulator back to the start, we get a new segment. So we get a new segment for each time we reset. And so at the end, we can iterate over these, and then the data contains all of this, again, is a block containing all these segments. So we can iterate over the segments, pull out the voltage, which was the first analog signal. Actually, this code's out of date. I need to change this slide. And then we plot it, giving some annotations for the axis, for the legend, and add some labels. And so what we end up with is something like this, where each of these traces comes from a different segment. And so the first trace is a hyperpolarizing current, the second one less so, the third one doesn't change at all, the fourth one we start to get some spikes, and then the highest current, obviously, we get more spikes. So Pine, the combination of Pine and Neo lets you kind of have this kind of multi-stage experiment and store all the data in a single object. As I said, Neo is kind of the center of a whole bunch of tools, so it can read data not only from simulators, but also from experimental data. And then, as I say, Pine generates Neo, and then Neo can be read or analyzed with tools like Elephant, stored in databases which are customized for Neo, visualized using various tools. So by using Pine and Neo, you're already part of this ecosystem of compatible tools. So we're getting quite close to the end. What's left now is more for people who are more advanced users. Perhaps you have your own simulator, or you know a simulator you'd like to use, but isn't supported by Pine. So I have a few slides. So if you were interested in adding a new backend for Pine, so each backend is a separate Python package. Those for Nest, Neuron, and Brian are distributed as part of the Python package, so you can get those for free, but you can also develop your own. So for example, the Spinnaker or the BrainScales implementations are distributed as separate packages, which you have to install, and those provide the Pine interface for that simulator. And so you can create this Python package any way you like. You can write it completely from scratch. So for example, the BrainScales version 1 implementation was written in C++, but with a Python wrapper around it. Or Pine does provide a kind of common implementation with some minimal hooks to make it easier to add new backends. You just implement these hooks, these certain functions, and then the common implementation takes care of all the rest. Or you can have some combination where you use part of the common implementation and part of it's written from scratch. So for example, BrainScales version 2 is this. It uses part of the common implementation, but also a lot of the code is written, of course, from scratch. And so, for example, a couple of years ago, the PyCal simulator came out, which is another simulator which has a Pine interface. But you don't get, this isn't installed by default with Pine. You have to go and download PyCal, and then you can start to use that simulator. But the same script should work with that as it does with Nest, Neuron, or Brian. So if you do want to use this common implementation, there's only a small number of functions you have to implement. So underscore create cells, underscore get parameters, and underscore set parameters for the population view, and then just conversion connect for projection. So projection implements all of these algorithms using only this basic conversion connect function. And then for the recorder, record, get spike timers, get all signals, and local count. So it's not too difficult to add a new backend. You also have to provide translations for the standard cell type. So from whatever the Pine name is, the Pine units, to whatever the parameters of your simulator are. So that's that. You may have heard of a tool called Sonata, which is an HDF5 based format for storing network structure. So sometimes if you've had a long, complicated algorithm to build your network, you don't want to recreate that every time. You just want to save that network to file and then load it in from file each time. Or you might have some other complex algorithm which determines which cell connects to which other cell. So for example, in the bluebrain project, where they're using neural models with dendrites, and they detect which dendrites are close or touch other dendrites, and they create synapses there. And so that's a very complex algorithm. And so then save the results to file, and just load them into their simulator when they want to run simulations. In fact, Sonata was originally a project developed by the bluebrain project and by the Allen Institute. So Pine also supports that. So if you've created your network in Pine, so you have some populations, you have some projections, you create a network object and you just export this to Sonata format. You can also take a Sonata dataset from elsewhere, and Sonata has two parts. One is the model, and one is the simulation plan. So saying, okay, well, how much current do I inject? How long do I run the simulation for? So you load these parts separately, and then you can execute your network model using the simulation plan and run the simulation like that. This will then save data in Sonata's own format, which you can then read into into Neo, using Neo, the Sonata IO format. So for anyone interested in that, that functionality is available. Getting a bit short in time, I'll very skip quickly through. There's a separate tutorial later today, I think, on music and on using music with Pine, so I won't go into that. I'll let Michael Dierfeld tell you about that. So I'll just say that, so Pine is open source, but not only is it open source, it's also a community project. So there's been 15, 20 people, I think, have contributed code to Pine over the years. We very much welcome contributions from anyone who's interested. So whether you found a bug, or if you want to add new features or new functionality to Pine, or really get involved in maintaining Pine and improving it, then please do come along. There is a developer's guide to help you get started. You can contact us on the eBrains chat or on the mailing list, which is in the wiki page. There's a link to the mailing list. So please do come along. Pine has been endorsed by the INCF as a community standard for simulator independent model specification. So we've demonstrated that we do have this functional development community, among other things. Yeah, and just to finish, I'd like to thank the various funding organizations, in particular the European Commission, which has funded Pine for many years through the Facets, Brainscales, and Human Brain Project. Also my organization, the CNRS, and a list of people who have contributed. This is possibly not completely up to date, but a lot of people have contributed to Pine over the years. If you have questions, you can find me on Twitter, or you can go to my website and find my email address. You're very welcome to contact me directly. And thank you very much. If you have any questions, I'm happy to take them.